Right, so this brings us then, I want to just talk a tiny bit about the development of the eye. And at its most basic, the eye really consists of a few cells sort of lying around and responding to light, basically. And then the eye gets a bit more complicated because it forms a cup. This is now a sophisticated sort of aid, and basically started life out of the salad bowl from Woolies, and then acquired through spontaneous sort of non-random mutilation. It lost its lid and it acquired a white form. And if I do this, and some of you are close by, you'll be able to see that there's light and dark. The edges are creating dark, and so you've got a kind of rudimentary eye. It can sense light and dark. It doesn't see very well, but as it drags a nautilus or some kind of primitive creature along with it, it can kind of function perhaps a little bit. The nautilus, of course, sees much worse than an octopus, and today the octopus sees about 100 times better, and it's got a fairly sophisticated eye, such as we'll see down over here. Darwin, of course, when he started to think about his ideas, was a little bit worried about the eye, because he kind of thought, well, this is a very complicated and very perfect type of structure, and did this fit in with his ideas? And then he had a few beers and he sat down and thought about it, and he thought, well, given enough time, the eye could actually evolve and get to where it needed to go. But basically and it's been estimated that to get from this very basic eye to an eye like a human eye or an octopus eye, you only really need about 400,000 to 500,000 years, which seems like a very long time, but when you think that the Earth has been around for 4.6 billion years, and life has been on the Earth for 3.5 billion years, and bacteria have been around for, 3 billion years, then 500,000 years is not actually so bad, and this will allow the eye to develop. In the most primitive form, this is already quite a sophisticated eye, because you can see here it's got a cup, it's got a little pinhole, because some of these primitive eyes, of course, would have been born with deeper cups or with narrower sort of openings, and occasionally there would have been a film across, and everybody would have laughed at the creature, but the film would have protected the eye, and would have been useful innovation or development in terms of creating more human or more the types of eyes that we're familiar with. This, of course, the octopus eye here, you can see is already pretty sophisticated. It's got an eyelid here, it's got a cornea, transparent cornea, it's got a lens which is more spherical in eyes, but it has muscles in the ciliary body here which can change this lens, and it's got an iris here which can sort of open and close the aperture and in that way regulate light. One of the interesting things about this particular type of eye is that the photoreceptors, the light-sensitive receptors, are near the front of the retina, whereas in our case, in the human eye and other vertebrate-type eyes, it's further back, it's closer to the pigmentary layer, and we'll come back to that a little later and what its significance is. All right, now here's the sort of simple sort of eyes, and of course you've got trilobites, which have been around for about from that early sort of Cambrian period, so around about 500 million years back. So these trilobites were living, and they like beetles, and they have these sort of insect-type compound eyes, which we're about to look at. And they've been around for about 500,000 years, 500 million years. Of course, the eye being soft doesn't preserve all that well, and so we don't have much of a record as we go further back in time. All right, so this is then the simplest sort of eye, this simple sort of compound eye. What's called a crystalline cornea is actually a lens. So what's happening here is the light is coming through the cornea here, through the lens, and the lens here is in apposition, it's touching the receptor here. And so, and an insect wouldn't have only one of these. They're going to have something like in the dragonfly about 28,000, and in the ant a few hundred of these types of eyes. So it's a compound type eye, and I'll show you a picture of it in a little while. But basically, it hasn't got a fantastic vision, but you can see here that the eye can pick up large objects and a little bit of detail here and there, and tell the difference between light and shades. And so for diurnal insects, it's pretty satisfactory, and for some of these other aquatic animals here. But in terms of the superimposition sort of compound eye, you've got a nice innovation, because some of these eyes would have had a space here between the sort of cornea and lens and the light-sensitive layer, basically. And what that means is that light from adjacent sort of facets or lenses can stimulate the same receptor, and that allows that receptor to collect more information and to allow the eye to have a better light resolution and light perception and so forth. And so nocturnal insects, for example, would have something like that type of eye. So this is then the typical sort of compound sort of eye. you have this creature here which is hasn't got its eyes even in its head. It's sitting on stalks, which is quite nice because occasionally something might eat the eye. And if you're lucky, the eye can regenerate. I'm not quite sure if the eye does regenerate, but if it can, then this is a useful sort of thing to have. One of the other nice things here is that this type of creature, as simple as it seems, it has 16 different types of color receptors, whereas we only have 3. So a far more sophisticated eye can sense polarized light, it can sense ultraviolet. They don't think it senses infrared, it's autofluorescence when it needs to, and it measures the wavelengths of light out there and sort of autofluorescence in relation to its environment. And so it's got a very sophisticated sort of light sensing system, basically. So you get all kinds of creatures, and although it seems as that there's this huge diversity across all these different organisms, you also need to keep in mind that there's commonalities, and most eyes will have a light sensitive layer. And there's certain genes like the PAX6 gene, which is a kind of master gene which controls eye morphogenesis and essentially where the position of the eye is in the head and so forth. And so it looks as if there's a huge diversity, and there is, but there's also a lot of commonality across species that seem very odd. And you'll find this PAX6 gene in mice, to the octopus, to humans, to fruit flies, and various other things as well. Spiders are quite interesting because they can range from having no eyes at all, they live in the dark with no light at all, to having only 2 eyes, to having 8 eyes. And they have here, you can see the 8 eyes here, and then here's a attractive creature with 2 over there and 2 over there, another 4 little ones over there, and you get squids which have eyes looking up and down and all sorts of strange things going out there, but never mind. Back to the spiders. So they've also got one quite useful sort of innovation as well. Besides being able to operate in all kinds of environments, they've got this layer of crystals behind the retina. And so light will pass through, stimulate the photoreceptors, hit this reflective layer, go back through the photoreceptors. And so, again, it helps them to attract or to accumulate more information and see a lot better. And this is, of course, why when you shine a light at a cat or a dog in the dark, you see this kind of reflective thing. It's because of the light hitting these crystals and being reflected back. You can do it with a human, but you need a very powerful light, and you have to hold it very close, and it's not very typical because we don't have one of these nice convenient layers of crystals that will work this way. All right, then we'll have to stop here. I'm kidding. Okay, when the retina, of course, is an incredible structure, and we don't really have time to get into it. But here, Cahal essentially did hundreds of drawings of different creatures and their nervous system, the retinas and the brain, and he used the Golgi sort of stain that was invented or discovered by this person here, Golgi, and essentially it's a silver nitrate stain, so it stains nervous tissue black here as against the yellow. And he already, each of these is a kind of little work of art, these hundreds of diagrams that he did, and that of course interests me with the art and the science and so forth. But he was able to identify already there's large and small ganglion cells, and there's receptors here, cones and rods, and had a very obviously fantastic idea of what these things were already and what they were for and so forth. And so the 2 of them eventually won the Nobel Prize for their work in improving our understanding of the central nervous system. Of course, today we can go to a website like this and we can see all kinds of healthy rats and not so healthy rats, and they also suffer from the same sort of eye disease as we do sometimes. When I qualified, you only had a light and a ophthalmoscope, you shone it in the eye, you looked at the retina. Now students, of course, have things like this. Here's the vitreous, this is the retina, this is our fovea, where when we look at an object, we direct our fovea towards that object. Not a healthy system. Here's this kind of pigmentary layer, this red or orangey. There are gaps. It's kind of got an elevation in the one part, and there's sort of spaces here, sort of cystic sort of spaces, and this is all irregular. So this eye wouldn't see very well, and there's some disease present basically in this eye. The choroid, which is sitting at the back here, of course, is kind of like a swamp full of blood. There's lots of blood vessels, and it's supplying nutrients to the receptor layer, which is sitting just above this pigment, the sort of orangey red part. We also have, we don't have one yet, a scanning laser ophthalmoscope, but optometrists and ophthalmologists all over the world are using them, and if the dean's got any money left over of them tonight, well, we'd like one. But especially one with an adaptive optic system like this, because you can see the resolution is a lot better, and we're actually seeing tiny little rods and cones, and we can diagnose disease. We don't see it quite as well as this, but you don't have to kind of remove the eye and prepare it and put it on a microscope slide, and patients tend to not like that much. All right, I mentioned there was this difference in the retina between vertebrates and invertebrates, and so something like an invertebrate without a sort of spine, essentially over here, their sort of receptors are near the front, and so the light comes here and it hits the receptors directly, whereas in our eye it has to travel through a whole lot of cells and neurons and all sorts of things before it actually gets to the receptor layer. And at first some people thought, well, this was a bad design basically. But of course it's important because of the nutrient supply, and things like birds will have a thing called a pectin, which is, they have an avascular retina, but they've got a pectin which is full of blood vessels, and it's sitting in the vitreous, and their receptors are closer to where this pectin is. And so it's basically relating to where the creature is and what kind of environment it lives in and how it's adapted to function effectively in that environment. Some examples of what we've been talking about, the retina with the fovea there, which was the diseased sort of fovea we looked at previously, the optic nerve. Here's this reflective thing that we see because of these crystals behind the retina which shines light back. Here's the pectin that has all these blood vessels and is supplying the sort of retina. Here's an eye with an owl. It's an owl, and it's been dilated. They put a drug in the eye to dilate the iris. And so you can see then you've got, there's this orangey part which is the iris. There's a cataract here, it's opaque. There's pigment and it's subluxated. So it's trauma, you experience some sort of trauma, and it's just showing you this sort of thing. Of course, birds are very interesting because they're more reliant on vision perhaps than we are even. If you can imagine an eagle, it's flying through the sky. When it swoops almost 200 or 300 kilometers per hour, you can see things at about 1 or 1.5 kilometers. About 4 times or 8 times better vision than we have. And of course, when this thing is swooping down on its prey and it's going at 200 to 300 kilometers per hour, one hasn't got much room for error. You don't want to be smashing into the ground at that kind of situation. How much time do I have? 15 so.